Now let's discuss Chapter 11, Section 2, the application of Mendel's principles. So we have to think about a little bit about math here and probability because genetics comes down to the figuring out the probability of certain events, certain combinations of things happening. And so as we know from flipping a coin, that you have a 1/2 chance of it being a head and a 1/2 chance of it being a tail. So any one flip of the coin gives you a 50% chance of one side or the other. Now, what is the probability of having 3 heads in a row? So we know one flip is half, another flip is half, third flip is a half, and the probability of 3 events, all with a 50% chance happening in a row, is essentially those 3 probabilities multiplied together. So you essentially have a 1/8 chance of flipping 3 heads in a row. And the probability of getting ahead on any one flip is not dependent on the other. That is, these are independent events. Or another way, the past outcome, what happened with this flip, does not influence what happens with the future one. And it's the same way with genetics. As we'll see, if parents have a child with a certain combination of genes, that does not preclude them from having that same combination or some other combination again in the future. It's all based on probability, and the past events do not influence the current or future events. All right, so in the first section, we talked about the law of segregation, this idea that individuals have two copies of each gene, and when they make gametes, they are going to pass on one of those 2 copies. Essentially, the 2 copies segregate or separate. So, for example, let's look here. We have these 2 tall pea plants, and their combination of genes is big T, little t. Okay, and it's the same for each of the 2 plants that are being crossed. So, again, this is the T gene for the height of the plant, and there's 2 forms of this gene, or 2 alleles. We have the big T allele, and we have the little t allele, okay? 2 forms of the same gene. And so when a particular plant makes gametes or reproductive cells, some of those gametes will get the big T, some will get the little t. You don't get 2 copies of the same gene in a gamete. Each gamete only gets 1 copy of each gene. And the probability or the proportion of gametes that will have a big T is 1/2, and the other half will have a little t. So if this plant makes a whole bunch of pollen grains, half the pollen grains will have a big T, half will have a little t. And let's just say this is the mom plant over here, and so they're making eggs. Again, 1/2 will have a big T, 1/2 will have the little t. All right, so the combinations you can get in the offspring, as we see here, you can get offspring that's big T, big T, big T, little t, big T from the mom plant, little t from the dad plant, and you can get 2 little t's, again, one from mom and one from dad. Well, what's the probability of those? Well, you have a 1/2 chance of getting a big T from dad and a 1/2 chance of getting a big T from mom. 1/2 times 1/2 is 1/4. You have a 1/4 chance of getting a big T from dad, little t from mom, etc., etc. You have a 1/4 chance for each of these. So the law of segregation tells us these 2 forms of the gene will separate or segregate, and there's a 1/2 chance of getting either one, and when you put those combinations together in the making of offspring, you multiply those combinations, those probabilities together. Now, of course, we see there's two ways of getting the heterozygous individuals. Again, you can get the big T from dad or the big T from mom. And so with those, you have to add those two probabilities together because there's two ways to get it. So you have a 1/2 chance of being heterozygous, 1/4 of being homozygous dominant, 1/4 of being homozygous recessive. So indeed, here's those terms. Homozygous, that's when you have both your copies are the same, either big T or little t. You can be homozygous dominant or homozygous recessive. Heterozygous is when you have two different forms of the gene, two different alleles. You're heterozygous. Don't find yourself saying heterozygous dominant or heterozygous recessive because that really doesn't make sense. You're just heterozygous. Adding dominant or recessive only applies to homozygous individuals. Now you can see up here when we cross these two plants, we can expect 1/4 homozygous dominant, 1/4 homozygous recessive, and 1/2 heterozygous. But if those two plants just crossed and had 4 offspring, are you necessarily going to get 1/4, 1/2, and 1/4? Not necessarily. I mean, it could be that by chance of those 4, they might all be homozygous dominant. But when do the actual ratios, what you get in the offspring, fit the expected ratios? These are the expected ratios right here, okay? When do the actual proportions of offspring match the expected? When is that? Well, when you have lots of kids, okay? So instead of just having 4, if they have 100, those two plants are crossed and they have 100 offspring, which is not unheard of in plants. Plants can have lots of offspring, basically. If you have 100, then you should get approximately 25, 50, and 25. You may not get exactly those numbers, but it will be pretty darn close. Does that make sense? Again, the more offspring you have, the more likely the actual ratios, the actual numbers will fit what you expect. All right, phenotype and genotype. So the phenotype is essentially the expression of the genes or how something looks. So when we look at the pea plants and the flowers, the phenotypes are purple and white, how they look. The genotype is the combination of genes that gives you a particular phenotype. So over here we have the possible genotypes for this P gene. You could be homozygous dominant, heterozygous, or homozygous recessive. And you'll notice that the homozygous dominant and the heterozygous individuals all have the purple phenotype, whereas the only individuals with the white phenotype are the homozygous recessive. So notice when you have this situation of a dominant allele and a recessive allele, you can have two different genotypes that give you the same phenotype. All right, and here again with the dominant recessive situation, you have one genotype that gives you this white phenotype. Also notice the ratios. If you do this cross, big P, big P, little p, here's what we get in the offspring. Okay. Notice that we get 3 purples to every 1 white, 3-to-1 ratio. And we get 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive, a 1 to 2 to 1 ratio. So notice our ratios of our phenotype ratio is different from our genotype ratio. That's not always the case, but it is in this particular cross. So, Punnett squares, we've sort of been doing those already. It's where you look at the possible outcomes of a particular cross. What you want to keep in mind is, so you've got parents here, these 2 heterozygous parents, and these are the types of gametes they're going to make, or reproductive cells. Remember that those reproductive cells are just going to get 1 copy of each gene. So when we put them in the Punnett square, this represents one of those copies or one gamete. Here's the other one. And here are the gametes of the other parent. And then you just fill in your Punnett square. And it's good practice, in my opinion, to put the capital letter or the dominant allele first. So there's our Punnett square filled in. And here you can again see our, if the big B is dominant, we'll assume here's our ones expressing the dominant phenotype, and here's our one expressing the recessive phenotype. Now on the right, we have the two-factor, or what we call the dihybrid cross. The one factor is known as a monohybrid, BRID. And here we've got two different genes, the T and the G gene. And so when you've got this combination, again, each gamete is going to get one copy of each gene. It's going to get one T and one G. We can get all possible combinations. We can get the big T with the big G with the little t with the big G, little t, little g. Those are our four possible combinations as you see here. All right, we fill in our Punnett square again, and also keep consistent on which side you put. Since we're putting the t's on the left side, always keep the t's on the left and the g's on the right. And when you fill in the square, the Punnett square, always keep your t's together and your g's together. That just makes it a little more orderly. Now here, with the dihybrid cross, of course, things are a little more complicated because we have two traits and we have a larger Punnett square. But notice again, if capital T and capital G are both the dominant forms, you'll notice these individuals here and here all have the same genotype. They are dominant for both traits. Notice here we have individuals who are recessive for the T gene, dominant for the G gene. Here we have individuals who are dominant for the T, recessive for the G, and are 1 out of 16 individuals that is recessive for both. So in these dihybrid crosses, when you're crossing two individuals that are heterozygous for both traits, you'll always get this 9:3 to 3:1 phenotype ratio. Okay. So when we contemplate these dihybrid crosses, that leads to what Mendel called the law of independent assortment. And what the law of independent assortment says is that when you have, again, these two different traits, like R and Y, those two different traits sort independently of each other. That is, you're going to get all possible combinations. And why is that? Well, it's because these two traits are essentially on different chromosomes, okay? And so when you make gametes, again, you're going to get all possible combinations, okay? You're going to get big R with big Y with little r with big Y, and little r with little y. They are sorting independently of each other because they are on separate chromosomes. Now, where things do not sort independently is when they are found on the same chromosome. So if the R and Y genes are on the same chromosomes, they tend to stick together when they segregate. But what Mendel was looking at with his pea plants were traits that were all found on different chromosomes, so they all sorted independently of each other. All right, so here are the principles that Mendel figured out from his studies with pea plants.